This is a typical scenario for Alzheimer's drug development, and we've seen a bit of this recently, where there is an exciting finding in the literature, and we all think that finally we might have cracked it. And then there's much enthusiasm, and the late, great Dave Sackett, who did a lot for evidence-based medicine, father of evidence-based medicine, articulated 200 different kinds of bias, and my favourite was hot stuff bias. And we get a lot of hot stuff bias in the Alzheimer's field, and then we get the dawning realization that actually this isn't what it, this doesn't turn out how it started off. And then we start again. And then when we start again, the same thing happens. The same thing happens. You know, history is condemned to repeat itself. The first time is tragedy, the second is farce, I think was Hegel on Marx. So why do Alzheimer's drugs keep failing? Well, an estimated 99.6% failure rate. Poor early detection methods make clinical trials difficult and costly. And here are some of the challenges: understanding pathophysiology and heterogeneity, diagnosis, the timing of treatment, the generalizability and translatability of our animal models, and importantly, the robustness and reliability of preclinical data taken forward. And it's the robustness and reliability of the preclinical data going forward that's the focus of Equipt, how we might try and fix that using Alzheimer's as an exemplar, but with much broader relevance across the neurodegenerative diseases and beyond. And I'm a stroke physician, neurologist really, and that's how we're another graveyard of drug development. So Equipt, the first IMI consortium completely dedicated to improving preclinical data, a joint undertaking from different players in the field, proof of concept in neuroscience and safety, facilitated by a quality management system, and then with an intention to expand it R&D wide if successful. So our vision is robust data and scientific rigor in animal studies with impact on the 3 Rs: reduction, refinement, replacement of animals in research, enhancing the pace of knowledge gain, and critically shortening the time needed to make new drug treatments available to patients. We'll talk a little bit about that later. So what's the issue with the quality of preclinical data? Well, here's this is from years ago now. This is Barta and colleagues in the Academy of Emergency Medicine. They went round at a conference like this, and they looked at the posters, and they said, what did the posters describe and how they did their research, and what did they find? And they found that if you looked at posters which didn't blind and didn't randomize, the odds ratio for them finding something interesting was 5.2 compared with those studies which did blind and did randomize. I'm interested in stroke. As I said, this is a candidate neuroprotective drug, NXY059, developed by Astra for stroke, and it went through a 5,000 patient clinical trial program. But when we went back after the clinical trial program fell over to look at the animal data which had supported that trial, 5,000 patients in the clinical trials, fewer than 500 animals in the published literature. That doesn't make sense. And although overall NXY is pretty good, it improves infarct volume by about 45%. In those studies which reported randomization, blinding during the experiment, and blinding in the assessment of outcome, that efficacy is substantially and significantly lower. So it's a problem if studies don't do things to reduce the risk of bias in the way that the studies are done. So how are we getting on? This is the prevalence of risks of bias in over 1,000 publications from leading UK institutions in 2009-10, whether they report randomization or blinding or inclusion-exclusion criteria or power calculations. These individual institutions are colour-coded to protect my future career opportunities, but there's a lovely Scottish blue colour here, and you can see this performance is shockingly poor. One paper out of over 1,000 did all 4 of those things, and 68% of them did none of those things. So with the benefit of that knowledge, then it's not altogether surprising that we've got a problem. And what happens if we look at the Alzheimer's disease literature in particular? This is our systematic, Kieran Egan's systematic review as part of his PhD of over 400 papers testing drugs in transgenic models of Alzheimer's disease, randomization in 16%, blinding in 22%, not one with a power calculation, and reported efficacy was higher in non-randomized, in non-blinded studies, as we would expect. So this is a genetic problem, it's a problem in the Alzheimer's literature, and we should be trying to do things about it. So first of all, we're in Equipt, we're looking at historical data from our pharma partners, from the literature, and using those to look to see whether we can find signals in the pharma data that similar things of randomization and blinding are in play. We have systematically developed guiding principles and criteria governing rigor in experimental design, conduct and analysis, and we did this by doing a systematic review of published guidelines, and we found 13,000 papers, of which 62 were relevant. We took 58 items, went through 2 rounds of Delphi, and then a consensus meeting, and I've got 33 items which there's general consensus are important for animal researchers to do, and we'll use those to feed into the rest of our work, into developing guidelines, along with the validation studies that I'm going to tell you about, and the historical data analysis. The Equipt quality system is going to support the essential processes, procedures and responsibilities to allow labs to implement the guiding principles. And this will comprise a toolbox with everything related to research quality that scientists may need to know about, created and maintained by us, a planning tool that would allow labs to work out which bits were most relevant to them, which were of the highest priority for them to implement. And this is something targeted not just at industry labs, but also at academic labs. And then a dossier where they put all their relevant information, almost like a trial site file, master study file that you would have in a clinical trial. So when someone comes along and says, how do you know that you do work of quality? You can say, here's my dossier, here's my evidence. And the idea being that rather than having a one-size-fits-all, there'll be a stepped, staged approach with some must-have core elements, and then as labs continue to improve, increase the rigor of what they do, they move higher and higher up this and can eventually claim, if you like, gold equipped quality status. And version 1 of that was rolled out earlier this summer. And now, another important thing that we've been doing is trying to work out why it is often that a finding that's made in one lab, when someone else tries to take it into their lab and replicate it, nothing happens. It's just flat. And this is a common experience, particularly of pharma trying to in-license findings from academic labs. So we've got actually 5 labs working together on open field and EEG and Irwin test. First of all, doing the experiments as they would normally do them themselves. That's done. Then, informed by our guidelines and by our issues on study design, trying to harmonize what they do across the labs to see if this will reduce inter-lab variability. This work's ongoing. And then after that harmonization stage, to test the system with ring testing of an unknown compound across the different labs where they're blinded to see whether they get the same results. And that work's just about to start. And finally, there's no point in having guidelines if people A, don't know they exist, or B, don't know what you mean by them. So we've got a whole training platform which we're developing to sustain the Equipt quality system over time. We've evaluated existing training modules, and here are the modules that are around for these different dimensions of what Equipt might be interested in. And we've just completed our 2nd summer school. So in terms of the assets that Equipt will produce, as well as being very interesting for the people involved, I think, and being helpful for academic researchers to see what is happening in industry labs and vice versa. And we've got PhD students who are spending some time in academia and some time in industry, which is good. We're going to end up with, firstly, a living systematic review identifying primary research in Alzheimer's disease. So one of the important things when you start research is trying to work out what's already known. And that's difficult when there's 28 million publications in PubMed. And the best way of doing it is through a systematic review. But if you do a systematic review of 28 million publications, it can take a while, as I know. So we've developed, working with James Thomas at the Institute of Education and Epicenter, we've developed a machine learning algorithm that will go to those PubMed records and will then extract from that, with a reasonably high degree of precision, primary research in Alzheimer's disease. And then we'll partition that research down into whether it's human research or animal research or cell culture research. And because you can run a PubMed search every night, this is continually updated and the machine runs through it. So we think we've identified about 26,000 primary research reports relating to Alzheimer's disease from an initial search of 450,000 PubMed returns. So we're very grateful that machine learning is a thing and it exists. And the idea is that at the end of the project, we'll just leave this running and present this information online so that someone who's interested in a particular drug, in a particular transgenic model with a particular outcome measured, can just go and click through and download a curated bibliography of the relevant publications up until last night's PubMed search. So that'd be a thing. I'd quite like that. We're going to have systematically created guidelines for the conduct of animal experiments, and this is important. Some of you, if anyone's been following what's been happening in the States, the NIH and NINDS had a meeting last year about the importance of providing educational materials for people in the life sciences. And they had a 2-day meeting where part of the blood on the carpet was because people couldn't agree what should be in the training curriculum. And now we've got a set of 33 things that we can show, we can demonstrate why we think they should be in the training curriculum. We'll have individual animal data from multi-site experiments, and these will be made available on Figshare or somewhere. We'll have an ontology for describing animal experiments. This is important because we've been putting together data from historical pharma data, from the literature, and from our ongoing experiments. We've had to work out a common language, a common ontology that will describe all of those things. Now, one of the problems with fair data, this is that data that we create in science should be findable, accessible, interoperable, and reliable, is the interoperability bit. If I've got an animal experiment in an Excel spreadsheet and you've got an animal experiment, it would be good if column B was always the sex of the animal. And so we've now got an ontology that will allow that. And we'll have a training platform and materials, including the materials from the 2 summer schools, and perhaps most importantly, we'll have the Equipt quality system, which will allow labs at the most basic level to just go online and self-certify that they do the basic things that are required. Basic things include having a quality manager, having a quality strategy and the like, but then to increase through the steps of that, through the stages of that, as time goes by, to improve further. So we're 2/3 of the way through. We're making good progress. It would be lovely to have much more time and much more money, but you can't get everything you want. Thank you very much for listening.